Good afternoon, everyone.
Good afternoon.
I would request the
Good
participants
afternoon.
from their friends that the session has already started, and please ask them to join. Okay, I would like to welcome our esteemed speaker, Professor Dr. Timir Borun Jha, on behalf of Maulana Azad College, and I would like to share my greetings with the participants once again. Before moving on to his lecture, here's an introduction. Though he does not need any introduction, but still, it's a formality and I have to do that. Professor Dr. Timir Boron Jha is a retired professor of botany in the West Bengal Senior Education Service, has served as head of the Department of Botany at Presidency College or University and Darjeeling Government College and coordinated major academic programs, including the Department of Biotechnology, DST, FIST, and DBT STAR schemes. He continues to work as an honorary teacher and researcher at Maulana Azad College, Kolkata. Born in 1954, he holds an MSc and PhD in botany from the University of Calcutta, specializing in cytogenetics and plant biotechnology. With over 4 decades of teaching and administrative experience, Professor Jha has held key roles, including Additional Director of Public Instructions uh, at the Higher Education Directorate, Government of West Bengal, and Officer on Special Duty at Presidency University. He has taught at multiple postgraduate departments and remains actively engaged in academic mentorship. His research contributions span plant biotechnology, protoplast culture, micropropagation, molecular cytogenetics, chromosome analysis, secondary metabolites, and DNA fingerprinting. Notably, he has established a plant chromosomal database for important crops such as lentil, capsicum, and rice, an achievement uncommon in India, especially from a college laboratory setting. He has guided 4 PhD students, evaluated numerous theses, and trained students and teachers across institutions. Professor Jha has authored over 95 research papers in national and international journals of repute. He has contributed book chapters and co-authored a variety of books. The book worth mentioning is Plant Tissue Culture Basic and Applied, which was published in 2016. He is an elected member of the Plant Tissue Culture Association of India and was nominated by INSA for international scientific collaboration with Japan in 2010. After such an illustrious introduction, I think all the participants should be eagerly waiting for his lecture. So, without much ado, I would like to invite Professor Cha once again formally to begin with his lecture. Sir, please do the honor and begin your lecture. Thank you.
Thank you. So, thank you. Good afternoon, all the participants and my fellow junior teachers who have come to attend or who are attending this refresher course online from various states of our country. So you can see that I have selected a topic which is molecular cytogenetics, concept and applications. And the picture behind this shows the helix of DNA and the chromosome structures. So during my discussion, whenever you will feel that you want to ask any question, you can ask me, you can interrupt me, there is no problem. That is the beauty of the lecture, that we have a participatory interaction. So why I have considered that molecular cytogenetics, not the classical cytogenetics, not the only cytogenetics, because the development of the molecular biology, development of the chromosome biology, everything gives us some opportunity that whether we can place this DNA, or we will go to details in the DNA structures, and we have the chromosomes, and only you are aware that these chromosomes are the mother structure of this. So if you see this again, this DNA, these are the chromosomes lying inside the cells, and the final day condensed chromosome structures. So why chromosomes are very important? I will consider 3 points before we start our story. Number one, that chromosome was coined this term in 1888, and you were aware that the structure of DNA helix was unveiled in 1953, and the fast genome sequence, particularly in plants, that is Arabidopsis, was reported in the year 2000. It means that chromosomes were known 7 decades before DNA discovery, 9 decades before gene cloning, and more than 11 decades before the genome sequencing of any crop. So that is the main point, that we were aware about the structure of chromosomes without knowing details of its genetic material or the structural components. And when we are considering this, we will want to see that how can we define the chromosomes. So when we have seen that chromosomes are coming from the genetic material, the DNA, so chromosomes are the discrete and condensed genomic structures that hold our genes. And chromosomes and genes represent different levels of genetic information and are complementary to each other. That means molecular biology, molecular genetics, genomics, and chromosomes are not different. They represent the different levels of genetic expression. History unveils that the term genome was coined in 1920 to combine the study of genes and chromosomes. Chromosomes were known, I have already told, and one very important observation is that of all these things, that means genes, DNA, and chromosomes, only chromosomes, not genes, are the only visible structure under a microscope. That means we can study chromosomes under a microscope in any laboratory. And over century, we are pursuing, or we are studying the chromosome structures of different plants and animals through our classical cytogenetics. But this classical cytogenetics, in case of plant system particularly, poses some problem. And you know that in the plant and animal system, if we consider the plant and animal system, then we will found that the difference lies there, the thick cell wall in the plant system, which complicate the preparation of the high quality chromosomes. In contrast, animal cells, where there is no cell wall, which is why the conventional method for preparing plant chromosomes uses a squash technique in classical cytogenetics. Those who are practicing or undergraduate or postgraduate courses where chromosomes are prepared, classical cytogenetics uses the technique, we call it squash technique. Whereas in animal chromosomes, this preparation, this squash preparation does not require, only because the presence and absence of the cell wall. In all, the organism's chromosome number is species specific. That means if we consider the plant kingdom, diversity is enormous. Enormous diversity is there in compare to the, if we consider the highest organism in animal system is the human being, so one species with 8 billion population. But in plants, the diversity is much more than that. And that's why, if we consider that Arabidopsis, which is the model system in plant and animal genetics, in one hand, it is a very small plant, 6 to 8 inch in height, it completes its life cycle within 6 to 8 weeks' time and produces viable seeds not less than 20,000. On the other hand, in the same plant kingdom, we have a plant, we call this Sequoiadendron gigantea, which can survive more than 2.5 thousand years. So, in terms of chromosomes, in plants, it ranges from 2 n equal to 4 to 2 n equal to more than 1200. 1200 in one plant, and that the name of the plant is Ophioglossum. And this is the largest number of chromosomes of any organism in the world. The fact that in these Ophioglossum cells, these chromosomes can accurately segregate this enormous number of chromosomes during mitosis is truly remarkable. So, if we consider, if you can see, this is the haplopappus gracilis only have the 4 chromosomes, 2n equal to 4 chromosomes. These are the haplopappus gracilis. This is our own work of Albuca virens or this is Albuca virens, where it is 2n equal to 6. You can count 1, 2, 3, 4, 5, 6. And this is Ophioglossum reticulatum, where more than 1200 chromosomes are there within a single cell. On the other hand, in animals, it ranges from uniquely from 2n equal to 2 in a primitive Australian ant, Myrmeca pilosula, to over 400 in some butterflies. And see, this paper was published in Science Journal in 1986, showing with the Jimsa banding of the animal, ant animal and chromosomes, which shows n equal to 1, that is haploid number is 1. But here is one unique situation is there. This is the female, this is the workers. In animal system, we call it the workers. So these males are haploid, as in other haplotypes, the somatic contains only a single chromosome. This is the male end containing 1 chromosome and the female containing the 2 chromosomes. So the minimum range is 2n equal to 2, but we haven't found this 2n equal to 2 chromosome number yet in any plant system. And what is the potentiality? This species is potentially of great importance in both laboratory and field studies on gene organization. So this is one important aspect why chromosome is important. And all of you know, or all of you heard the name of Drosophila. How the chromosome looks like? This is the looks of the chromosome in Drosophila. We call it polytene chromosomes. And here, the genome contains 4 pairs of chromosomes. That means 2n equal to 8. The chromosome looks like this. This has the chromocenter, and the XX, the sex differentiation is there, XX and XY pair, and 3 pairs of autosome. The whole genome contains 15,682 genes. So that means genes and chromosomes are nothing but the different levels of expression. And this is again very interesting that in some Drosophila, we have another type of chromosomes, we call it B chromosomes. You can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 B chromosomes has been reported in Drosophila chromosomes. These are the accessory chromosomes, not the normal chromosomes. So whenever we are studying chromosomes, these amazing features we can make visible to our students. It was previously believed in case of what happens in the case of human being. We are all aware that we human beings have 46 chromosomes. But previously it was believed that the human beings like chimpanzee, gorillas have 24 pairs of chromosomes. But after the discovery of DNA, that is in 1955, Geo and Levan, using improved techniques, confirmed that humans actually have 23 pairs of chromosomes. So, where the additional pair gone? Additionally, it was discovered that rather than a pair of F chromosomes being missing in humans, 2 F chromosomes have fused together in our genome. Specifically, chromosome pair 2, the second largest human chromosome, is formed by the fusion of two medium sized ape chromosomes. So you can understand why chromosomes are very important till nowadays. So this is a normal chromosomal plate of human karyotype showing this 46 chromosomes. So now, what is our objective in chromosome preparation? That high quality chromosome preparation with a distinct morphological configuration is a prerequisite for determining chromosome number and karyotype analysis in plants and animals. So, what was the problem in classical cytogenetics? The classical cytogenetics has some problems, and that is the visualizations of the chromosomes. That means the cells contains the cell wall, the cells contain the high cytoplasms. So, in the modern method, this we have been able to eliminate both the cell walls and cytoplasms from the background of the plant cells, and we can make the chromosomes visible. That means this visualization of chromosome is heavily dependent on how the tissue or structure is prepared for microscopy. And you are aware for plant somatic chromosome studies, we always use preferably the root apical meristems or root tips. For many decades, chromosome preparation through classical cytogenetics in plants has enormously benefited the taxonomy, phylogeny, evolution, genetics, breeding, and biotechnology. However, the progress in molecular biology or molecular genetics has highlighted the limitations of classical cytogenetics. Nevertheless, the development of chromosome preparation technique in the 1970s revitalized the significance of chromosomes, ushering classical cytogenetics into the era of molecular cytogenetics. So that is why I keep the title of this presentation as molecular cytogenetics. That means from the era of classical cytogenetics, we have entered in the era of molecular cytogenetics to reveal some molecular features under the microscope to our students and researchers. So, the basic molecular cytogenetics approach is known as the enzymatic maceration and air drying method. Simply, I can tell you that this enzymatic maceration and air drying method helps the elimination of the cell wall. That means enzymes can digest the cell wall, and maceration eliminates the cytoplasmic part. That means through this EMA method, we can have a preparation where you can see the very distinct morphology of the chromosomes. So this evolution enabled the preparation of high quality chromosomes free from cell walls and cytoplasm, making the use of cover glasses optional because you are aware in cytological classes in conventional method we always use this obligatory use of the cover glasses and allowing the reuse of the same metaphysics for different experiments. But when we are not using any cover glass, so that means we can use the same slide for different type of experiment. And what are the different type of experiments? That means the same slide can be destained and restained with fluorescent stains and even fluorescence in situ hybridization. So, and karyotype analysis using molecular cytogenetics offers valuable insights by directly identifying DNA-based specific AT and GC heterochromatic regions that consist of repetitive sequence on chromosomes. That means the relationship is focused again through the chromosome visualization under a microscope that we can pinpoint some heterochromatic regions within the chromosomes under the microscope, which was rarely possible or impossible through our classical cytogenetics method. Molecular cytogenetics has greatly advanced further in the identification of the individual chromosomes, particularly through the use of non fluorescent and fluorescent stains, as well as techniques like fluorescent in situ hybridization, which we call FISH, genomic in situ hybridization, and spectral karyotyping. But for the limitation of time, I will restrict my discussion, especially on fluorescent, up to the fluorescent stain application in plants and also in animal systems. So, through the advancement of molecular biology, what we have come to know that the genome have revealed that every genome contains repetitive DNA sequence. You can see this picture where you can find the euchromatin and heterochromatin. And if you recall your postgraduate days, so it was Hage in 1938 who for the first time pointed out, considering the stainability, that the chromosomes contain some heterochromatin and euchromatin regions. So this is the molecular view of the heterochromatin and euchromatin region. And whenever we are looking the molecular view, we can see the repetition of the genomic sequences. We call it heterochromatin. So there are heterochromatin zones throughout the whole chromosomes. And these repetitive DNAs, commonly known as repeats, form the structural backbone of chromosomes. That means chromosomes having the repetitive sequences, but through classical methods, we cannot reveal the position of any repeated sequences on the chromosomes, which is very important not only for our crop improvement, but also for the knowledge of our students. And these repeats represent the most rapidly evolving sequences within a genome. So, whenever we learn through our molecular biology and molecular genetics that the genomic sequence contains repetitive DNA and these repetitive sequences are part of the chromosomes, our efforts was whether we can visualize these repetitive sequences under the microscope. Their amplification, what does these repetitive sequences? Their amplification and loss on a genome-wide scale can significantly impact karyotypes and the overall genomic composition. Chromosomal studies thus becomes crucial for examining the distribution and evolution of these sequences, especially given the challenges associated with analyzing sequence data. Because analyzing sequence data is very much challenging, so this can be an alternative, this can be an complementary if we can look the repetitive sequences on the chromosomes under a microscope. So now we have to know, we have to come that repetitive sequences within a genome serves as a valuable molecular marker if we consider that what is the purpose of these repetitive sequences and how we can utilize this. So at the chromosomal level, we consider the repetitive sequence within a genome serve as a valuable molecular marker for analyzing genetic diversity. Among whom? In the plant system, between the two species, within the cultivars of a single species, that means intraspecific and interspecific diversity detection becomes easier if we consider molecular cytogenetics as our base experimental system. It often exhibits specific genomic profiles related to particular species or cultivars. It can be used as cytogenetic probe to study chromosomal organization in plants, as karyotyping markers, and to reveal both the inter and intraspecific genomic diversity. Additionally, understanding the karyotype diversity can shed light on how species adapt to environmental changes. That means whenever you are preparing a chromosome through molecular cytogenetics method, that is enzymatic maceration and air drying, and you are looking a distinct morphology of the chromosomes, you can measure their length. You can say what is the total length, that is the TCL, total chromatin length. And when you compare the total chromatin length of different cultivars, you may get understanding the nature of diversity happening in the nature. So, molecular cytogenetics is an effective method for detecting DNA based specific repetitive sequences at various locations within chromosome under a microscope. So, we must have to know that at the molecular level how the chromosome looks like. If we consider this is a chromosome, the metaphase chromosome, so you can see this is the telomere. We call it the telomere. All you are know aware about it. This is the centromere or primary constriction. This is the repetitive sequences. They may be present the repetitive sequences, and this whole DNA where the repetitive sequences are present, we can call it the satellite DNA. This is the satellite DNA, that means repetitive sequences throughout the chromosomal region. That means repetitive DNA, how many types it may be, repetitive DNA in plant genomes can be categorized into two broad groups. One is dispersed mobile elements and tandem repeats. Tandem repeats includes ribosomal DNA and satellite DNA. Ribosomal DNA, all you are aware, that is in plants, it is the region of the secondary constriction. Ribosomal DNA comprises the 45 base rDNA are conserved in the cells of eukaryotic organism. That means this is a conserved feature. So if we get some variation, we can compare that as a marker. So these conserved rDNAs are located at the inwards of the secondary constriction region and comprises 18S, 5.8S, 28S rDNA genes plus transcribed and untranscribed spacer region, we call it ITS. And satellite DNAs are typically found in distinct loci on the chromosome, especially in the centromeric, in the telomeric, and interstitial chromosomal regions of the genome. So now you can look how a molecular chromosome can looks like. So this is the conventional and molecular features of chromosome. You can see this is the red, this is the centromeric tandem repeats, blue is the telomeric repeats, yellow is the subtelomeric repeats, green is the intercalary tandem repeats, brown is the dispersed repeats, and white genes and low copy sequences. So this is a normal chromosome. This is where we won't find any of these features, but through molecular cytogenetics, we can visualize some of these region which can act as chromosomal marker or cytological marker or for diversity analysis. So that is why this is very important steps or very important type where we should have some uh, training, or we should learn or we should transfer our knowledge to our students. So this is the I'm not going into again the nuclear genome in the I have told this. So how we can apply this in the plant system? Primarily, I'm considering the plants because plants provide everything to us. You are aware that from plants we can get food, we can get clothes, we can get money, so everything. So, how can we use this molecular cytogenetics in the plants? This is the lentil. All you know that lentil is very important in Bengal. It is called mushur dal. It is a uh, pulse crop, and this high protein content, and it is the in our diet. It is the everyday diet. And this chromosome, this chromosome prepared through the classical method. I will show you what is the difference when we are applying the molecular cytogenetics. This particular lentil contains 14 chromosomes, 2n equal to 14. So this is a very good plate in the classical cytological method, but in all karyotype, we call it karyotype, that means the total number of chromosomes, the representation of the chromosome, we call it also karyotype. So this karyotype, every karyotype must have one pair of secondary constriction. And where may be the secondary constriction? This secondary constriction I will show in other species, that it may be in the terminal region or in the interstitial region. But in the classes, we never find that this where the secondary constriction lies in this particular plant species. So this is, you can see the background, is the cytoplasm, it is also stained, chromosomes are also stained, and this is the EMA based preparation where there is no cytoplasm, only you can see the chromosome morphology very distinctly. And you can see that these two chromosomes, these arrow marked chromosomes, has some gaps. So we find some gaps when we stain these EMA based chromosomes with the GIMSA. And remember, this GIMSA was used first in making karyotypes of animal systems. As animal systems do not have the cell wall, so the animal scientists for making the human karyotypes always use GIMSA. But in case of plants, application of GIMSA started from 1917 onwards. So now, this same plate, whenever we can go for molecular cytogenetics, what we can do, we can destain this plate. We can destain this plate, and the same plate can come for the molecular fluorescence staining. And fluorescence staining are always, we consider two fluorescent dye. One we call DAPI, 4,6-diamidino 2-phenylindole, which is AT positive. Wherever there is any repetitive sequence, AT-rich repetitive sequence, DAPI will target that sequence and shows its visibility. So we can identify that these chromosomal regions have the AT-rich. And on the other hand, there is another fluorescent stain, we call it CMA, chromomycin A3, which is just reverse. It attracts the GC-rich regions. So that's why in molecular cytogenetics, we generally use these two contrasting nucleic acid specific dye. These are nucleic acid specific dye, and they attach to the either AT-rich or GC-rich regions. So now you look in some cultivars of the lentil, some cultivars, not in all cultivars, we obtain so many these AT-rich regions whenever we stain the chromosomes with DAPI. You can see that this is another cultivar showing number of different regions with, and intensity of stain also varies. We can mark these are the different DAPI region which was not possible through our conventional method. And when we make the preparation with CMA, you can see this too, this is the secondary constriction region. So it means that these secondary constriction regions, which was not even visible in the classical Orsin method, it lies interstitially, not in the terminal region, but interstitially. So that is the beauty of the molecular cytogenetics, that for our students, we can clearly show them the molecular look of the chromosomes, and we can find where this NOR region lies, whether it is interstitially or it is terminal. So we are sure that students will be benefited, you will be enriched to see this method through which we can pinpoint or we can mark cultivar specific AT-rich region or the GC-rich regions within any karyotype. So I will, in the next slide, I will show you that the lentil wild species, there are some lentil wild species. I collected it from the pulse research station Kanpur, and where we obtained that in the wild species, the secondary constriction lies terminally. So you can see this is the terminal secondary constriction. We have pinpointed for the first time that this is the lens lamoti and this is the lens nigricans. Both are wild type, and this wild Indian species of the lentil contains the terminal secondary constriction instead of the interstitial secondary constriction present in all the cultivated cultivars. So remember, this can help you in the future breeding programs because we know that as a gene pool, these wild species are always possess very important genes which are not transferred to our cultivated cultivars during the transition or during the breeding method. So we can consider that which gene we want to transfer, and through chromosome analysis, we can also check that whether that has come to the uh, resulted cultivar or not. So these are the list of our publications in lentil. And this is the capsicum. You all know that India is a rich repository of capsicum, and uh, you will astonish to know that for the first time we have made the database, a fluorescent database of these Indian capsicums. And if anyone from the northeast regions or North Bengal, they know that this is the highest pungent capsicum. We call it Bhuj Jalokia or Naga Jalokia, and we have done their chromosomes, and they have the three species. One is annuum, one is frutescens, and one is chinense. Chinense, this Bhuj Jalokia belongs to the species chinense, and we call it the very small type of capsicum. We call it dhani lonka. This comes under the species of frutescens. That all have the 24 chromosomes. So, what is the way to differentiate them? So, we can differentiate them if we go through the process of molecular cytogenetics. And one very interesting thing we noted and we reported for the first time that who are in North Bengal and northeastern region, there is one capsicum called Dale Khursani. So it is in the hills for many decades, but nobody checked its chromosome number. So how chromosome number benefits us? When we check the chromosome number, we are surprised to see that they have 48 chromosomes instead of 24. So we never discriminate. We consider, yes, this is Dalicursani is a type of capsicum, like the similar capsicum, but it carrying 48 chromosome instead of 24. So this is very important discovery through the chromosome analysis. So now if we can see, this is the capsicum annuum. So you can see the very good plates, and you can see these are the secondary constriction, that is the terminal one. So can we confirm the terminal one? This is the same plate. So look, when we call it with the DAPI, you can see these regions have make dull appearance. That means they are not GC rich region, they are not, there is no AT rich regions, so they become dull. Look, this is this one and this one. So this one and this one, they have become dull. And the same chromosome, you can see this one. So in the CMS staining, they are showing positive signal. That means the secondary constriction lies in the terminal region of the chromosomes. Besides that, we have got some more signals in DAPI and CMA. And this is the capsicum frutescens. This is the same 24 plates, but we can get variation at the level of fluorescent banding. And this is the world's number one pungent capsicum, where we have found that this is, you can see, this is the terminal secondary constriction, and you can see this is not the AT positive side, this is not the AT positive side, but it is the GC positive side. So we can consider this method as very valuable for identification and characterization of plant chromosomes. So these are the Bhujalokia. These are the chromosomes of Bhujalokia. So in the same chromosomes, we have obtained the secondary constriction and the chromosome morphology. So you can check the difference in the chromosome morphology also. And now the story of the Dali Khursani. And this capsicum has recently, just 4 or 5 years ago, this has given the GI tag. And GI tag means it is more valuable now. And we collected different, you can see this is the leaf size. Leaf size of the capsicum is much more than our normal diploid capsicum. So this is the polyploid capsicum leaf size, this is the diploid capsicum's leaf size. And these are the different Dale Kursani types collected from different parts of West Bengal. So what is the chromosomal status? And this capsicum, why we are considering this important? Because capsicum has been growing local the Sikkim and Darjeeling hill districts for many decades without its genetic status being known. This capsicum is very pungent and locals call the capsicum Dali Kursani. Cultivated polyploid capsicum is a rarity. There was no report that in the capsicum any polyploidy exists except one wild species. However, we have for the first time reported the existence of the cultivated allopolyploid capsicum with 2n equal to 4x equal to 48 chromosomes. It is behaving like 2n, but actually its chromosome number is 48 and 4x is the basic chromosome number. That means it is divisible by 4. That means 12. That means it has an allopolyploid. That means two species of crossing between the two species. We do not know which species they are, whether the parental species, but through molecular cytogenetics, if we go ahead with the fish, we can uh, provide the genomic characterization of this capsicum. So Dali Khushani has received the DI tag from the government of India. So look, these are the 48 chromosomes of the different Dali Khushani, and even we get the fluorescent signal in the 48 chromosome plates. Very interesting, very interesting. So look, why chromosome analysis is important. It gives the real identity at the genetic level. It is not possible through any other methods. That means through DNA isolation, through genomics, we cannot say whether this is a diploid or triploid or polyploid. So that is why this molecular cytogenetics helps us. Classical cytogenetics also for number, but molecular cytogenetics gives us added advantage that we can go through to locate the heterochromatic zones, secondary constrictions, some molecular markers. Very important. These are very important for these are our publications. And now, last 3 or 4 years, we are concentrating ourselves in the rice. Rice is a major staple food crop consumed by nearly half of the world's population. It belongs to the family, grass family, that is Poaceae, and comprises two cultivated and 24 wild species. The major cultivated Asian rice which we consume, that is Oryza sativa, India ranks first globally in total rice cultivation area and second as a producer. And its chromosome number, you can see that this is 2n equal to 24. So the capsicum was 2n equal to 24. Rice is 2n equal to 24. So unless we analyze their chromosome, how can we know that the water, the differences at the chromosome level? And even in the rice, they have the wild species, either they are diploid or tetraploid, will 2n equal to 4x equal to 48. So unless you analyze, unless you reveal what is the status of a particular species, we cannot say anything. That means whenever we are carrying out our molecular analysis work, chromosomal interpretation act as complementary information. So look, You have seen the chromosomes of lentil, you have seen the chromosomes of capsicum, and you can see the magnification. This is the 5 micron magnification. That means more than two chromosomes comes within the 5 microns. And as we have done, and this is the first time in India we have established or we have standardized the method of molecular cytogenetics for rice chromosome analysis. And look, these two chromosomes, very small chromosomes, but showing some apical portions, which we consider this is the secondary constriction. And how we are confirming in these small chromosomes? You can see the DAPI of the same chromosomes, it has shortened. That means this part is not showing prominently, not showing visible stain. So we can confirm that this is 80 negative regions. That means this is not the 80 rich region, this is DAPI negative regions. That means this is the GC, probably this will be the GC rich regions. So in molecular cytogenetics, this is one GI tag rice. This is black rice from Manipur. This is a GI tag rice. I will show you some GI tag rice chromosome. This is Tulai Panji from West Bengal, India. This is also a GI tag. And you can see our preparation provides ample location of the secondary constriction in rice. And we have checked that the difference in the number of chromosomes, that means which chromosome pair bears the secondary constriction, we have obtained the diversity in chromosome number bearing or carrying the secondary constriction in different cultivars of the rice. This is one important findings through molecular cytogenetics. This is another, the Govindaho. This is another GI tag rice. So this is, you can see the secondary constriction lies here. And here we, in all the cultivars, one similarity is there, that the secondary constriction is terminal. We have completed the karyotype analysis for more than 90 Indian traditional and wild species cultivars. And now this few slides will provide some molecular thinking for all of you, because this is one species with the B chromosomes of aloe vera. You know, this is the aloe vera, the very important medicinal plants, but in the undergraduate and postgraduate class, this is also a model material for chromosome analysis. And every time we train the students through classical cytogenetics method, it contains 14 chromosomes. And if you divide the 14 chromosomes according to their length, you can find 1, 2, 3, 4, 5, 6, 7, 8. So 8 long chromosomes and 1, 2, 3, 4, 5, 6. 6 small chromosomes. This is the classical Orsin staining. So you can tell me, sir, this is a well scattered metaphase plate, no problem in counting of the chromosomes, but counting is not enough. What we have learned from our previous slides, that we have learned that the chromosomes contain repetitive the sequences. So we want to see whether repetitive sequences are present in the aloe vera chromosomes or not. So we perform the EMA technique. So you can see the difference lies here. The stainability, the morphological configuration, it's much more clear. And you, in this plate, it is very difficult to identify or to locate the secondary constriction position, but you can see in the Jimsa stain plate, we can see, we can mark this is the secondary constriction plate, and some length heterogeneity also present in the aloe vera chromosomes. We call it bimodal karyotype. And bimodal karyotype means the size variation, or it is divided into two distinct groups. That means 8 long chromosomes and 6 short chromosomes. That's why we call it bimodal karyotype. So, very interestingly and surprisingly, When we stain the same plate, you can see this is the same plate we have stained with DAPI. What you can look here, just see, this is the primary, all these are the primary constriction. If you consider this is the primary constriction of this chromosome, the second one, so this is the primary constriction of the second one. You can see this has taken DAPI positive signal, showing DAPI positive signal. And you can find this 1, 2, 3, 4, 5, 6. All the 6 small chromosomes in their primary constriction region, they are showing DAPI positive signal. And out of the remaining 8, you can see 1, 2, 3, 4. Out of the 8, 4 chromosomes, primary constrictions of the 4 long chromosomes showing DAPI positive signals. Altogether, in the karyotype, we have identified primary constrictions of 10 chromosomes showing DAPI positive signals. On the other hand, in the same karyotype, we didn't find any DAPI positivity in 4 chromosomes. So now question arises. Why this is so. Within a species, not within a species, within a karyotype, we are getting variation which was really impossible to see through our classical cytogenetics. So now, if you go back to think that what is the gene composition of the primary constriction and what is the function or purpose of the primary constriction. So this question naturally come to our mind, that what is the function of the primary constriction. All you are aware, in the cell division, we have studied, we have learned, we have teaches that primary constriction is the site where spindle fibers attaches. So this is very conserved function, evolutionary conserved function. And this generally, in many plant species, we have seen, or in some animal species, that these primary constriction sites are generally AT-rich regions. It has been reported in many cases. So if it is AT-rich region, so 10 chromosomes are showing AT-rich signal. Then why 4 chromosomes are not showing any AT positive or DAPI positive signal? So can't you question that we should go to analyze the molecular sequence of this primary constriction region? So this is amazing that though it is very conserved function, but its genetic diversity, sequence diversity is present not only within the species, not only within, it should be not only within the karyotype, but it is almost chromosome specific. So how within a cell, all the chromosomes behaving accurately during the time of cell division, having diversity in primary constriction regions, genetic diversity in the primary constriction regions. And this type of genetic diversity has been reported in rice and in many other cases. But what we have achieved in this very simple plates, we can raise the question, what is the gene sequence? What is the repetitive sequence present in these primary constriction regions? Can't we be? Can't we raise these questions? So only when we are looking the chromosomes under the microscope, we can raise this type of questions for our next research. So this is one very important observation of this lecture. And in the next slide, you'll be astonished to see the next slide. This is the same aloe vera slide. You can see in all the plates, we have got more than 100 plates. We have studied more than 100 plates in aloe vera. as it is very popular, very important, and very classical material for students at the undergraduate and postgraduate classes. This is another material, we call it methi, the Trigonella phenum gracium, which has 2 n equal to 16 small chromosomes. Here also, we could not locate the secondary constriction. But when we come to this point, we can find that the secondary constriction here is present in interstitial region. So students can learn through this method the what is the actual position of the secondary constriction, and secondary constriction are always called a landmark position of any particular species. And what happens in when we studied these cells with the CMA stain, is a very remarkable just contrast in contrasting the reverse situation what we have seen in the aloe vera. So this is the same plate. You can see the same plate. So this is the Giemsa stain. You can see this is the DAPI stain. This is the primary constriction region. This is the primary constriction, primary constriction. All the primary constrictions of the Trigonella shows GC reach regions or CMA positive zones in the primary constriction regions. So now you compare that in aloe vera, we are we have noted 10 out of 14 chromosomes showing DAPI rich region, DAPI positive signals. And in Trigonella, in another plant species, all the chromosomes, that means 16 chromosomes, their primary constrictions are not AT rich but GC rich. So, this is another very unique observation. This is another very important class material. So, whenever we are just changing the procedure, whenever we are considering the different methodology and applying the stains and the same slides, what I told you at the beginning of my lecture, that we can reuse the same slide. That means you can make stain with Gimsa, you can distain it, you can stain with DAPI, you can distain it, you can stain with CMA, you can distain it. If you have facilities, you can go for fish with this slide. So these are very important observation why we need to learn this technique, why we need to transfer this knowledge to our students who are the future of our country. And now the very common material, that is the allium cepa. This allium cepa has 2 n equal to 16. So Trigonella has 2 n equal to 16, allium cepa is 2 n equal to 16. Unless we do it, how can we know that allium cepa chromosomes are much bigger than the Trigonella? And what we got in allium cepa, that in when we stain the same plate with the CMA, all the terminal regions fluoresces with CMA stains, all the terminal regions of the chromosomes. That means at the beginning, when we dissected the chromosomes at the molecular level, we told that the repetitive sequence may be in the terminal, subterminal, primary constriction, secondary constriction. That means on the all satellite DNA regions. So this is another instance when we can show our students that these terminal regions carry the uh, repetitive sequence and which are GC rich regions. Just reverse the same one. This is the allium sativum, which you call roshun, garlic. So allium sativum, we could not find a single GC rich or AT rich region through DAPI or CLA. So this is again raises the questions about the sequences of this uh, allium sativum uh, gene sequence or repetitive sequence, that why they are behaving so, what is their nature. So is they DAPI neutral or CMA neutral? It appears that they are DAPI neutral or CMA neutral. What we achieve from this allium sativum, that they possess either one pair of secondary constriction, or three secondary constriction, or four secondary constriction for the first time in any Indian cultivars. So you can see in one root we have got the two secondary constriction regions which are interstitial. But in Allium cepa, if you see, this is the secondary constriction. This is the secondary constriction. This is the secondary constriction. But in Allium cepa, sativum, we got the interstitial secondary constriction. So there is a difference between, though they have the 20 equal to 16 chromosomes, but at the level of landmark chromosome marker, that the secondary constriction, they may be two, they may be three, they may be four, without disturbing their overall morphological features or other features. But at the chromosome level, we have checked, or we can tell that this species requires molecular analysis at the molecular system or genomic system or any other system to find out what type of gene sequence they are carrying. This paper is downloadable. This paper has been published in Karyology, and as a teacher, I can say that this will help enormously to our students. And one objective of my lecture is that to instill the interest on plant chromosome research, because as I am from the University of Calcutta is the seat where chromosome research started in 1946 with Professor A. late Arun Kumar Sharma, who is my mentor also. This is BCA fava, and this is one species which is shown with only 6 chromosomes. So we can see that this chromosome morphology can be very clear cut. This is the Orsin stain, this is the DAPI stain, and this is the CMA stain chromosomes. You have seen some sort of small chromosomes, but this is the smallest chromosomes, smallest than the rice chromosomes also. And for the first time, this is Amaranthus cruentus, having 2n equal to 34, very small chromosomes, uh, probably more, some chromosomes are more smaller than rice chromosomes. And why I'm showing this, for the first time we have noticed the terminal primary constriction. That means telocentric chromosomes. So if you can see, this is one telocentric chromosome, this is one telocentric chromosome, this is one telocentric chromosome, this is the CMA stain, this is telocentric chromosome. We obtain 10 telocentric chromosomes within the karyotype. So the species, we have shown you that the centromere may be present in the metacentric, it may be submetacentric, even it may be telocentric. And this amaranthus is an example where we can get, where we can show our students the telocentric nature of the chromosomes. So finally, in plant system, I can show you, or I can tell you, that molecular cytogenetics can be applied irrespective of the chromosome size. Irrespective of the chromosome size, that means starting from aloe vera, the big chromosomes, allium cepa, the big chromosomes, to the Amaranthus cruentus and rice chromosomes. And this is one rice wild species showing 2n equal to 40, 2n equal to 4x equal to 48 chromosomes. So this is all about our application in plant system for molecular cytogenetics. So if you want, we can have a break of 5 minutes so that we can have a discussion, and then we briefly summarize of the karyotype preparation and importance in human being. So if you have any question, you can ask me the question now. Any question?
Uh, Hello, sir. There is a question from Dr. Namrata Ray.
Yeah.
She asks, sorry, from Sonali Ray. She asks, can molecular cytogenetics differentiate T clones? Can molecular cytogenetics differentiate T clones?
Differentiate?
T clones, T. Cha, cha.
Cha, T.
T clones, yeah.
Actually, in T, it's chromosome T. That means Camellia sinensis. Is it the question on T?
Yes, yes, sir. Yes, sir.
Oh, so you know that the T contains the chromosome number 2 n equal to 30, right? So all the Camellia sinensis, all the species, irrespective of its different cultivar, the Chinense or Ashamica, all contains 2 n equal to 30 chromosomes. So nobody has attempted this molecular cytogenetics works in India. So if we can initiate the work on T, it will definitely, I am sure, we will get differences at the molecular level when we will study the molecular cytogenetics and chromosome karyotypes. Definitely, there should be some difference. Definitely.
Thank you, sir.
Okay, sir. If any other participant wants to interact with sir, you may please unmute yourself and proceed with your question.
Hello.
Yes,
Uh-huh.
Sir, I wanted to ask that in
tell your
all the
name.
slides,
Tell your name.
my name is Arunima,
Yeah,
Arunima Datta. Sir, in all the slides, we have seen that you have experimented on different plant species.
yeah,
So now my question is, if we can use CMA staining, DAPI, and EMA GIMSA to
not CMA
identify
GIMSA, not EMA GIMSA. Yes, yes.
EMA GIMSA, EMA GIMSA, then can we predict which kind of chromosomes in which type of animals, because I am from zoology background,
Yeah,
can be more prone to translocation or breakage, because
yeah,
more number of rich regions
yeah.
can have a, probability of breaking of more. So maybe that can lead to certain studies where we can pre-predict certain
Yes,
normal
absolutely.
conditions.
Because in the next few minutes, I will discuss some aspects of the karyotype preparation of human chromosomes.
Okay.
So when we will discuss the human chromosome preparation, though the same procedure can be applied for any animal system,
Okay.
right? This type of animal, cat or whatever you like. So if you can establish and if you can check with the DAPI and CMA, and even in animal chromosome, if you have the laboratory facilities, go for fish.
Yes, sir.
I will tell a few words about fish, but really it will be very exciting. Really, it will be very exciting.
Yeah. Thank you, sir.
Oh, okay. Any other participant, if you have any question? So I think there are no more questions.
So
That's
I
all.
will continue.
Yeah, yeah.
Let's continue. Okay. So now the preparation method. So at the very beginning, I told you that the basic difference lies with the plant and animal is the presence and absence of cell wall. So in human being, what generally is done, blood is considered the most frequently used sample tissue. But cultured skin fibroblast or bone marrow cells are also used for this purpose. But blood is taking sample blood is easier. So, and we all you are aware that not all the blood type cells can divide frequently. Only none of the leukocytes in the blood normally divide, but lymphocytes can readily be induced to proliferate, providing a very accessible source of metaphase cells. So in human being, not only blood, only the lymphocytes. That means whenever you are culturing the blood cells, you will get the division of the lymphocytes. You have to increase the number of the lymphocytes. Lymphocytes are round cells that contain a single large round nucleus. So we can separate this, the red blood cells, the white blood cells, and plasma. So we can take the lymphocytes as a initial material, which will be helpful, which are regularly doing in any animal system laboratory for preparing the karyotype. And you will be astonished to know in West Bengal, there is not a single lab where you can do this human karyotype. Whenever you are asking them to perform the karyotype, they're sending your slide to elsewhere. And it is very costly also. So my intention to encourage the younger teachers, so think about this type of things where skill is required, where training is required, where we have adequate number of students, so we can teach the students, we can make them trained in different systems so that we can have a trained number of students. So a sample of blood is drawn and coagulation is prevented by the addition of heparin, what is regularly done in our laboratories. So mononuclear cells, lymphocytes and monocytes are cultured for 3 to 4 days in the presence of a mitogen like phytohemagglutinin, which stimulates the lymphocytes to proliferate madly. That means tremendously. The lymphocytes are harvested and treated with a drug such as colchicine. Colchicine is like the colchicine, which you use in plant system. And the purpose of the using colchicine is that it disrupts mitotic spindle and prevents completion of the mitosis and passes through a hypotonic solution. This makes the nuclei swell osmotically. In case of plant system, we use this hypotonic solution. We use the fixative, but here we use the hypotonic solution. The Solent cells are fixed, dropped onto a microscopic slide and dried. So this is much easier than the plant system, because we have in plant system we have to eliminate the cell wall through enzymatic digestion, and then we have to macerate either with a fine tip tweezers or with the squash procedures. But in case of animal systems, as there is no cell wall, so the drops can be put on the slides and it will spread on the slide, and after drying, we can stain them. The slides are stained with Giemsa. So Giemsa staining was initiated in animal system, not in plant first. But in plant, as we have the so much diversity, so now we are regularly using Giemsa in molecular basic molecular cytogenetics work. So they are scanned then to identify good chromosome spreads. That is, the chromosomes are not too long or too compact and are not overlapping. And in animal system, there are karyotypic software is available. So you can photograph them and you can be cut and pasted using the computer. That means if you use this photograph software, so they will make the homologous chromosomes group together. So if standard staining was used, the orderly arrangement would be limited to grouping like sized chromosomes in pairs. So this is the steps how we can perform the human karyotype in our laboratory. This will be useful for not only our students but for our teachers also. So look, this is the normal gene sustain plate. This is the normal gene sustain plate, and this is the normal human karyotype showing the sex chromosomes. And this is the chromosome classification of chromosome karyotyping. So we can make the karyotyping in this way. And if we use the Giemsa with different method, so you can use Giemsa banding. So within a human system, how many bands are coming? And with counting the band, we can differentiate between the two human beings, whether there is any variability or not. And the best application of human karyotype provides a comprehensive overview of an individual's chromosome stability. It provides the chromosome stability, and all you are aware that genetic diseases are, though low in number, but they are severe in nature. So this karyotype preparation may act as valuable tool for identifying the genetic abnormalities in the individuals where directly chromosomes are involved. So I am citing three examples. One is Down syndrome. All you are aware that the Down syndrome is induced through the trisomy of the chromosome number 21. So if we can make a karyotype from the patient, and if we find that the three chromosomes, 21 number chromosomes are present in three number, so we are sure it is giving us 2n equal to 45 plus xy. So it is signaling that the patient has a Down syndrome and counseling, genetic counseling is required for that. So again, another disease is the Turner syndrome, where the instead of XX or XY, it comes as the X0. And this means one's sex chromosome is less. That means it has 2n equal to 44 chromosomes. So different, that means monosomy Turner syndrome. And this is Klinefelter syndrome, where sex chromosome becomes one extra more. That means 2n equal to 44 plus XXY. That means through the human karyotyping, at the very basic level, we can not only counsel, we can determine the patient's stability or individual stability at the chromosome level, and we can say them whether they are suffering from any these type of genetic diseases which are involving the whole chromosome number. And the same chromosome preparations can be done for fluorescence in situ hybridization. And fluorescence in situ hybridization is a technique that uses DNA probes. That means the short DNA sequence designed to hybridize to a specific region of the genome, that means with the chromosome. So hybridization between DNA and the chromosome to detect complementary sequence on the chromosomes. So you can understand this is very important experimental setup where you can detect the genes, specific genes. It is undertaken in situ on chromosomes, spread on a slide and visualized under a microscope. So to find out a particular gene with the help of the DNA probe on the chromosomes, it is now also now possible, and it is done over more than 3 decades. And fish can provide information on both the copy number of the chromosomal region, whether it is deleted or duplicated, and the chromosomal location of a particular region, whether it is subject to a structural rearrangement such as inversion or translocation. And the example is that Cry-Duchat syndrome involves the deletion of a portion of the chromosome 5. So if we perform the fish experiment and if we find that the deletion of chromosome number 5 has taken and the patient morphology is giving the same observation that this is a genetic disease related with the chromosome number 5. And Philadelphia chromosome associated with chronic myelogenous leukemia, a result of reciprocal translocation between chromosome 9 and 22. So these are the very important thing in molecular cytogenetics. So look, this is the fish showing the dicentric fragment, fragment, dicentric, fragment. So we can detect the fragmentation of the chromosome, the addition of the chromosome. And the next slide, you can see the telomeric signals in the human chromosomes. So all the telomeric signals, this type of observation is now possible. Not only that, we can multicolor fish. That means at a time, more than one probe and more than one stain we can use to make the chromosome multicolored karyotype in human chromosomes is now possible. And this is the distribution of the 5S rRNA genes on the chromosome 1 45S RNA genes in human chromosome 13, 14, 15, 21, and 22. That means 15, 21, 22, 14, and 13. So at the gene level, we can use this fluorescence fish technique for identifying or locating the genes, and we can compare with the normal persons for this. And this is the spectral karyotyping. Is a molecular cytogenetic techniques used to simultaneously visualize all the pairs of chromosomes in an organism in different colors. So all these are now under control. So finally, chromosome analysis through molecular cytogenetics offer a cost-effective method for determining unique species-specific chromosome number and their morphological configurations. The analysis can reveal the distribution and characteristics of repetitive DNA in both primary and secondary constriction regions, as well as in satellite DNA regions. Such information is fundamental and complementary, and it can be challenging to obtain from next generation sequencing data. That means this type of next generation sequencing cannot provide the foundational information of the genomic structures. Since chromosomes and genes represent different levels of genetic information, karyotype analysis can uncover valuable genomic details that will never become obsolete. It is important to note that karyotype analysis should not be seen as an alternative to DNA sequencing. That means we never consider that chromosome is an alternative to DNA sequencing. Not at all. Chromosome informations are always complementary. They have the distinct yet complementary roles in genetic research. Chromosome preparation protocols can be conducted in any laboratory without the need for specialized equipment, and they yield robust result while being cost effective. So this is fundamental type of research. It does not require much investment. It does not require so much high cost, high throughput instruments, but can be set up at the college level or university level. Cytogenetics provide a conceptual foundation for modern genomics because unless we know, unless we see the morphological features of the chromosome, the numerical numbers of the chromosome within a species, so it is a half-hearted work if we go for genomics. So chromosome at the base level, we must have some knowledge that how many chromosomes are there, what is their length, how they looks like, where are the secondary constrictions present. With this information, if we go for molecular genetic analysis, genomics, that will be better. In the context of climate change, genome biology encourages a closer collaboration among the field of genomics, cytogenetics, molecular biology, and bioinformatics. Compiling the karyotype diversity of cultivated and wild crops is important for enhancing the gene pool because all we are aware that the human population is increasing and we have to feed 10 billion population in 2050. So keeping in mind, we have to increase our production. So for increase of production, we have a collaborative approach. We shouldn't have the separatist approach of separating chromosomes from genome biology, separating chromosomes from bioinformatics. So all together we can have a better information data set for cultivated and wild crops because these wild crops are providing the important gene pools for future crop improvement. So in human beings, it served as a kit for prima facie detection of chromosome-induced genetic diseases. The number of researchers interested in cytogenetics is steadily decreasing worldwide. It is essential to revitalize cytogenetics training. We must inspire students by providing them with the incredible experience of observing the remarkable structure of chromosomes under microscope. Understanding chromosome structure is crucial and we need to continue developing high throughput cytogenetic approaches to keep pace with the rapidly advancing field of genome morphology. And my objective of today's lecture is to inspire the young faculties to look this portion of the biology for future generation. Thanks to you all. Thank you.
Thank you, sir, for such an interesting presentation. We got to know about the difference between conventional cytogenetics and molecular cytogenetics. Attending your lecture was a deja vu kind of an experience where I felt like it was my personal experience, like returning back to our PG classes. And I hope that all the participants must have benefited. Now, I would like to request them to post their questions in the chat box, or if they want, they can interact with Sir directly.
Yes.
Okay, sir, there is a question from Dr. Rakhi Chakraborty. She
Yeah.
asks, secondary constrictions are mostly located terminally or subterminally.
Yeah.
Is there any significance of this position?
Yes, because it is species specific. Normally, the position of the secondary constriction is species specific. So, in any species, that means whenever we studied in lens, this in lens it is interstitial. That means not terminal. And in case of aloe vera, it is terminal. In case of allium, it is terminal. So this terminal region or the interstitial region, it is a landmark chromosome marker for any particular species.
So we have another question from Dr. Shobhik Mitra.
Yeah.
He asks, some literature suggests the presence of M chromosomes in some bryophytes. Are they accessory chromosomes like the B chromosomes?
M chromosome.
Yes, yes, sir.
Actually, there is still some controversy when we advance in our molecular biology work. So, how they are functioning, the B chromosome or M chromosome, it is not fully known. So sometimes we call it the B chromosomes are without any constriction. And one concept is that acentric chromosome cannot survive. And we have seen that B chromosomes are always not present in all the cells. It may be present in some cells. It may be present in reproductive cells, not in the somatic cells. So these type of variations are there. This type of problems are there. So anyone can work with this type of problem. There is no bar. So these are very interesting field to take these challenges through the molecular cytogenetics method with the help of this nucleic acid fluorescence dye or the fish technique. If we can perform this type of consider specific that B chromosomes are present in the somatic cells of this species. So if we carry out this fluorescence technique, so we can see whether they are actually taking any fluorescent dye or not. So whether there is any heterochromatic sequences are present or not. And regarding the heterochromatic sequence, they are not totally unraveled. Their function is not totally unraveled because these are the sequences which have deposited within the particular species during the process of evolution. So we have got the total chromatin length variation. So this total chromatin length variation does not mean their chromosomes are different. Their chromosomes are same, but some amount of heterochromatin, how they have deposited, we do not know. But they have deposited during the time of evolution. So this process can help us to understand the chromosomes and molecular aspects of the genes.
Okay, sir. Thanks for your explanation. Anyone else? If you want, you may interact with Sir directly.
Thank you everybody for
Okay.
patient sharing. Thank you.
Mm, mm.
Thank you. You
Okay.
also.
Well, we are ending this session. And before we move on to the next session, I would express my gratitude to Dr. Timi Bharunja once again. And I would request the participants to stay online for our next speaker. He
Okay.
has joined. Okay. And
Thank
I
you.
would like to invite Dr. Dipon Udhikari for conducting the next session. Thank you. Over to Dr. Dipon Udhikari.
A very good afternoon to one and all present over here. I'm Dr. Dipon Udhikari, Associate Professor of Botany Department of Maulana Azad College. Let me take the pride in to introduce before you our next speaker, who has already joined, Professor Nagaraj Balasubramaniam. Just a minute. It's not open. Yeah. Professor Nagaraj Balasubramaniam, who has already joined our team. So before starting the session, I welcome Dr. Nagaraj in our uh, open session, and allow me to give a brief idea about his work and his achievements. A cell biologist, Dr. Nagaraj is a biologist by training and graduated from the University of Mumbai with a bachelor's and master's degree in microbiology. He worked in the Cancer Research Institute, now ACTREC, at Tata Memorial Center for his PhD in biochemistry. He took a break from cancer biology to do a postdoc in mammalian phototransduction at the University of Miami, only to return to cell and cancer biology, focusing on cell adhesion and trafficking as a postdoc first and then a research assistant professor in the lab of Dr. Martin Schwartz at the University of Virginia. He moved to Azad, Pune to start his lab in the year 2010. He has got research interests, including the understanding how cell adhesion regulates the trafficking and its complication in cell migration and anchorage dependence. He's holding the current position as a professor of biology department in Azad, Pune. Over to Dr. Nagaraj for his next lecture.
Can
Thanks a
you hear
lot.
me?
Yes, I can hear you,
Can you
sir.
hear me? Okay. I'm going to
Uh,
share my screen and you can tell me if it's visible. Just a minute.
yeah,
Hold on.
sure, sir.
Mm.
Yeah, it's visible, sir.
Is it, can you see this?
Yes, we can see. Hope
Okay,
the
sorry. One
participants
second.
also are
Yes, sir, we can.
they're okay. And you can start, sir. It's perfect.
My
Thanks.
presentation is visible?
Yes, your presentation
Yes,
is
sir.
Okay,
visible.
okay, wonderful.
Yes, sir.
Thank you. Thank you for the invitation to be here. And I think I apologize. I have a cold I'm recovering from, so my voice isn't very great. Um, and I will try and tell you, a story that has taken many years for us to put together. I think I have a little bit, time here to talk about how scientific discoveries evolve, and how they eventually lead to the prospect of things like treatment. I think there is a lot of emphasis today on developing treatment strategies, and a lot of that begins in basic science. One of these, this story essentially also tells us how that happens, and that's what I'm hoping we'll be able to communicate to you. So I'm talking to you from this campus in Pune, the ISER Pune campus. At the onset, I wanted to acknowledge the fact that my lab has funding from many sources, government as well as private sources, which has allowed for us to be able to do the work that we are doing. And so without this kind of funding, we will not be able to pursue the questions that we are pursuing. My lab works on cell matrix adhesion, and I'll kind of use a couple of slides that are very basic. These are slides that normally I use when I'm talking to undergraduate students at times. But this kind of sets you up for the questions that we are asking, and sets you up for thinking about why this is relevant. Cell matrix addition is an interesting idea in cells, and when you talk about cell addition, you essentially mean the ability of cells to stick or become attached to something, and this they are able to attach to either cells to other cells or to the extracellular matrix. A classic example of cell adhesion stems from the fact that we all develop from this ball of cells which are held together to create complex tissues and organs, and a lot of that is mediated by the fact that cells are able to stick to not just other cells but are also able to stick to this matrix that is secreted by these cells in their extracellular space, and that matrix then drives, not only keeps cells together, but also drives the behavior and functioning of cells per se. So when I think about this and when I'm describing this to students, for me, the matrix reminds me a lot of, a bowl of Maggie. I think we've all had a bowl of Maggie, and, if you've been absent-minded like me, occasionally you have forgotten a bowl of Maggie. And if you forget a bowl of Maggie, it kind of forms this nice gel of Maggie. If you take the bowl and invert it now and remove the bowl, you have this structure that mimics the shape of the bowl, and the Maggie there is essentially what the extracellular matrix looks like. And if you imagine all the vegetables as being cells, they are held in this matrix, which is the Maggie of multicellular life, that allows them to come together and work together as a unit. A couple of years ago, we wrote this article that looks at the matrix from from the perspective of students, where we brought together ideas of how the matrix is put together. And if you're interested, you're welcome to go check this out, right? So it's published in this public science magazine called I Wonder, and if you scan this QR code, you should be able to download this article. This introduces you to the idea of how the matrix is assembled and how it is put together. This is where the lab, the thinking of the lab begins. We've been thinking about how this matrix influences cells. And this is an interesting combination because the matrix, it works both ways. As in the cells obviously secrete the matrix, but the matrix also comes back to influence the cells. So there is this feedback loop that is going both ways. So my lab works on cell matrix adhesion, and we've been interested in different aspects of cell matrix adhesion. We've been interested in how it regulates trafficking, membrane trafficking, endocytosis, and exocytosis. And a good part of the lab is also interested in organelle function, that I will not be talking about today, but if you're interested, do reach out to us, if you're interested in that aspect. A small part of the lab also works on looking at cell matrix cell implant interactions as well, right? A good part of the work that I'm going to talk to you today about focuses on looking at this pathway of exocytosis. And I'll tell you a little bit as we go along on why I chose to talk about this today per se. So as I said, the matrix is something that the cells secrete. It's this Maggie of life that comes back to influence cellular behavior, and it does this by many different ways. Everything from anchorage to being a migration track, a signal reservoir, and also importantly, helping cells respond to biomechanical cues are things that the extracellular matrix does. The matrix also is interesting because it regulates the ability of cells to work with growth factors to regulate anchorage dependent signaling, which essentially means that if cells are attached to the matrix, their ability, the way the cell responds to growth factors that they see is going to be very different from if they were not attached. And this ability of adhesion to regulate growth factor signaling is what's also is what also confers anchorage dependence in cells. Now, many years ago, there is, a long time ago, there was this idea that in the relationship between cell surface area and anchorage dependence of growth, suggesting that how cell surface area may contribute to this anchorage dependence. Now, anchorage dependence is interesting because it's an important regulatory paradigm. It also ensures that this is what drives the behavior of quote unquote normal cells, right? And it becomes relevant when you consider the fact that just as normal cells are anchorage dependent, there are disease conditions like cancers where the cells become anchorage independent, right? So understanding anchorage dependence is relevant also because of the fact that, it allows you to eventually understand how diseases like cancer could occur, right? So this idea of cell surface area regulating anchorage dependence is something that I explored as a postdoc in the lab of Martin Schwartz, where we essentially looked at the role of how adhesion could regulate membrane trafficking. Now, this seems like a very intuitive idea, but at the time when this was proposed, it was fairly radical, and nobody had really looked at this association or this crosstalk. And this summarizes a very elegant piece of work that eventually got published in Science in 2004. And it looked at something very simple but seminal. That is, if you take cells which are adherent and you detach them, it looked at the way the plasma membrane organization changes. And what they noticed is that if you take cells and put them in suspension, and if you label the plasma membrane, there is this very rapid endocytosis that takes place of lipids from the plasma membrane. They go into the cell and they stay there as long as the cells are non-adherent, right? So you see this nice red dot inside of the lipids that have, come and accumulated, and they stay there as long as the cells are non-adherent. So the plasma membrane pretty much gets cleared of these lipids, which essentially means the plasma membrane loses all these signaling lipid platforms that exist. This leads to a turning off of adhesion-dependent signaling on loss of adhesion, and so the signaling that is turned on and stays on when the cells are adherent is now lost on loss of adhesion, right? So that's a very interesting regulatory change, and we spent a lot of time evaluating how this process works, right? Like what actually drives endocytosis, what drives exocytosis. And what we discovered is that on loss of adhesion, the endocytosis that happens through caveolae, right? Caveolae are these membrane invaginations that are present on the plasma membrane. They're interestingly responding to not just biochemical cues, but we later discovered they are also responding to biophysical cues when the cells are detached. And once that endocytosis happens, that bright internal spot of lipid that you saw, we discovered is actually the recycling endosome. And from the recycling endosome, the return back to the plasma membrane of these lipids is again under the control of adhesion through this RAL-R6 pathway that works along the exocyst complex to deliver stuff back to the plasma membrane, right? So this endocytosis and exocytosis balance seems to determine the amount of these lipid domains on the plasma membrane, which then regulates adhesion-dependent signaling. So what this also led us to speculate and then eventually show is that if you want, so in a normal cell, the loss of adhesion triggers this endocytosis, and on loss of adhesion, the exocytic pathway is turned off, because of which stuff comes in, does not go back out, and stays in this perinuclear recycling endosome pool, causing the plasma membrane order and composition to be altered and signaling to be switched off, right? So if you want to achieve, if this is the mechanism by which anchorage dependence is controlled, and if you want to achieve anchorage independence, right, then the best way to do this would be to disrupt one or more of these. You could either, what you're really trying to achieve is keep these lipid domains on the plasma membrane, keeping signaling on, and you could do this either by blocking endocytosis or by stimulating exocytosis. So you could do either of this, right? There is a block of endocytosis, or there is an activation of RAL exocyst dependent exocytosis. And remarkably, both of these things happen in cancer, right? So in cancer cells, caveolin is lost as a tumor suppressor. And as I will tell you in much detail today, the activation of RAL, in cancer cells drives the RAL exocyst complex to keep this exocytic pathway on. The net outcome of this is, by blocking endocytosis and triggering exocytosis, the lipid composition of the plasma membrane in these cancer cells is the lipid composition is maintained, as even when the cells are non-adherent. And so in a normal cell, when you detach cells and put them in suspension, the turning off of signaling that happens does not now happen in cancer cells, right? So we discovered what was a very fundamental mechanism of regulating anchorage independence. Now, the advantage of making such an observation is that now you had a whole bunch of questions to ask on how these processes are regulated, and what exactly happens in cancers, right? So among the things that we decided to look at, which I will talk to you in some detail today about, is to look at how the adhesion-dependent regulation of RAL is mediated, and asking how that controls, how that regulation is affected, if at all, in cancer cells, right? Our objective in the long run was very simple. We had a mechanism that we had discovered is playing an important role in regulating anchorage dependence in normal cells. We knew that this mechanism can be deregulated in anchorage independent cancer cells. If we figured out how this process works, right, and the mechanism, the players, the regulation, our hope was, can we target these pathways in anchorage independent cancer cells and see if we can bring them and make them more anchorage dependent, right? And if we are able to achieve that, then in principle, we will have another mechanism or a tool by which to target cancer cells, right? And disrupt their anchorage independent growth. This anchorage independence is what allows cancer cells to be able to go to new places and establish themselves, in processes like metastasis. So the story I'm going to try and tell you today is going to begin with our understanding of how adhesion regulates RAL activation. How did we go about looking for this? What exactly did we discover? What did it tell us about the regulation of RAL? And importantly, what new avenues of regulation did it open? How that allowed us to go look for new regulators of RAL, which brought us to the role this kinase called Aurora kinase could have in regulating RAL activation. And eventually how, using the Aurora RAL pathway, we were able to develop a method to target this signaling in anchorage-independent cancers to try and suppress their anchorage-independent growth, right? Which is what we, began with as our objective right at the beginning. So I'll introduce you to a little bit about what we discovered in the process. Among the first important discoveries that we made was that RAL has two distinct isoforms, RAL-A and RAL-B, shown here. They are very similar, but they have this C-terminal tail, which is distinctly different. And we had discovered that adhesion is able to regulate the activation only of one of these isoforms, RAL-A, but not RAL-B. That suggested that downstream of adhesion and in the anchorage independent pathway, one RAL is more important than the other. This reflects in if you target RAL, how spreading is affected by RAL-A knockdown, and not so much by RAL-B knockdown, right? What this that then led us to ask is importantly, how could this regulation be happening, right? So we began by asking whether if adhesion is able to regulate RAL-A and RAL-B, does that is that mediated by how adhesion regulates different RAL gaps, right? Now, RAL gaps are essentially proteins that regulate activation of small GTPases, in this case RAL, and there is a whole family of them. And interestingly, there is a group which is RAS dependent, and there is a group which is RAS independent as regulators. RAL is a small GTPase, which means its activation is dependent on this GTP-GDP loading cycle. So when it is GTP bound, it's active. When it is GDP bound, it's inactive. And that activation is controlled by these gaps. The inactivation is controlled by gaps. So what we did is, we did a simple screen looking at all the gaps, knocking them down and asking which GIF, if at all, is able to regulate the adhesion-dependent spreading of RAL. Now, why did we look at adhesion-dependent spreading? Because this we knew to begin with was mediated by RAL. And so if there is an effect of targeting of one or more of these gaps, it's very likely that the gap that we are that is having an effect is possibly regulating RAL. And remarkably, we found that there is one GIF, which is this RGL1, which if you target, regulates adhesion-dependent spreading. This we did using siRNA smart pulls and individual siRNA knockdowns. And knowing that that is the case, we also extended this study to ask what happens in caveolin null cells. And the reason we did caveolin null cells is, at the beginning, I told you how endocytosis happens through caveolin or caveolae. And so if you knock down caveolin, you essentially compromise the endocytic pathway that is then regulated by the exocytosis. So what we knew is that if the effect of this RGL1 knockdown is happening through RAL and is happening through the exocyst pathway, it only works if the endocytic pathway is functional. So if you don't have the endocytic pathway, which is triggered by loss of adhesion, which is what happens on loss of caveolin in caveolin null cells, you lose this regulation. And the fact that we lose that regulation in caveolin null cells told us that, it's likely that what we are looking at is an RGL-mediated regulation of RAL-A. So we then asked, if you knock down RGL1, does it do something to RAL activation, RAL-A versus RAL-B activation? And we are able to pull down active forms of these protein using a very simple assay where we take a GST tagged fragment of a protein, which is an effector for RAL. And so you attach this to a bead and you pull down, RAL that is able to bind to this GST tagged fusion protein. And you can probe for RAL-A or RAL-B and detect how much RAL-A or RAL-B has been brought down. And remarkably, what we saw is that if you target RGL1, the amount of RAL-A that is brought down drops significantly without affecting RAL-B. And this happens in the first 10 minutes of cell spreading, suggesting that this is an adhesion-dependent event. This happens with the smart pools that I showed you here. It also happens with the individual siRNAs. And when we are using individual siRNAs, because these target very specifically RGL1, we can also do reconstitution. That is, make a mutant of RGL1 that cannot be targeted by this siRNA. And when we put that mutant, which is shown here by this asterisk, you can see that the regulation, this effect on RAL activation is restored, right? Suggesting that indeed it is dependent on RGL1. So we then did another very simple but elegant experiment where we said to prove that it is regulating only RAL-A but not RAL-B was rather important, largely because these are very similar proteins. And the question was, why should it regulate one but not the other, right? So what we did is that we took this constitutively active mutant of RAL, which is the RAL V23 mutant. We also compared it to the RAL-B constitutively active V23 mutant. And we saw that if you take the RGL1 knockdown cells and add back active RAL-A or active RAL-B, only RAL-A is able to restore the spreading that we see, but not RAL-B, suggesting that, it's RAL-A that is being regulated and so is able to bring stuff back, right? What we also did further, which is a very interesting twist to this experiment, is that, we made these constructs where the tail of RAL-A and RAL-B, which is different, we switched. Okay. Now, the tail of these RAL-A and RAL-B is very vital to their localization in cells. So it determines where A goes and where B goes. And our hypothesis was the reason RGL1 is able to regulate RAL-A here is largely to do with where RAL-A is sitting relative to RAL-B. It doesn't have to do with RGL1 recognizing something unique in RAL-A as compared to RAL-B, because they are not that unique. They are very similar. It's but for the fact that their localizations are different. So we made this tail switch mutant. And what we interestingly now see is that when you switch the tails, active RAL-B which now has a tail of RAL-A and therefore goes to where RAL-A sits, is able to restore function, while RAL-A, which has a tail of RAL-B and now goes to where RAL-B was sitting, is not able to, right? Which was really interesting and kind of made the point very elegantly that this is about RGL1 sitting with RAL at a very specific location, allowing for this regulation to take place, right? And so this told us that there is a clear mechanism for how RGL1 can regulate RAL-A activation in early adhesion. We also wanted to check, early adhesion is an integrin-dependent event. What really happens in late adhesion, right? This is when cells are nicely adherent, polarized, and migrating. Is the regulation any different at all? And to do that, what we did is we looked at RGL1 knockdowns, and interestingly, in late adherent migrating cells, where they're allowed to migrate in the presence of serum, what we noticed is that targeting of RGL1 does not regulate RAL-A activation, but it in turn regulates RAL-B, right? Which suggests that now, in response to cells being adherent and polarized, and now seeing serum growth factors, it's the regulation, possibly the localization of RAL-B and RAL-A is altered in such a way that now RGL1 is able to regulate RAL-B activation and not RAL-A. And this is reflected in the fact that if you knock down RGL1, its behavior, in terms of the way migration happens, is comparable to RAL-B, where the migration is actually faster. So the red and the yellow are RGL1 and RAL-B. The control and the blue and green are control and RAL-A. And so RGL1 now in stable adherent cells, which are migrating, does something completely different in regulating RAL-B but not RAL-A. Right. So early adhesion, RAL-A, not RAL-B. Late adhesion, RAL-B, but not RAL-A. Right. This kind of gave us this very interesting pathway on how adhesion in early adhesion in the presence of low serum conditions could regulate RGL1 to regulate RAL-A and therefore affect cell spreading. Now, there is a player that I've introduced here in this pathway that we will talk a little bit about, which is Aurora kinase A. And I'll tell you a little bit about why we are thinking about Aurora kinase A, right? I'll come back to this slide. What we also did, and I'll briefly allude to this, that we looked at cancer cells and we noticed that in normal cells, just as RGL1 plays a role, in cancer cells, RGL1 could play a role depending upon its relative levels with other RAL-GEFs. So we looked at two different cancer cell types, MIAPACA2 cells and SW620 cells, which come from very different origins. And one has less RGL, more RAL-GDS. The other one has comparable levels of RGL1 and RAL-GDS. And what we noticed is that when you knock down RGL1 in these conditions, there is hardly any effect on RAL-A or RAL-B activity. RAL-GDS seems to affect both RAL-A and RAL-B. And when RGL1 and RAL-GDS are both present in comparable amounts, now RGL1 is able to regulate RAL-A but not RAL-B, and RAL-GDS is able to regulate RAL-A and RAL-B together. Right. So there are some very interesting regulatory controls that are operational here. It suggests that RGL1 could choose to regulate a certain RAL. Unlike certain other GEFs like RAL-GDS, which, no matter when you are looking at them, their regulation seems to be lacking the selectivity that RGL1 has, right? So that's the kind of brief summary of what I have told you so far, that how downstream of adhesion this pathway could work, and how RAS signaling could differentially affect both of these, right? We went on to look at other regulatory players and discovered that, RAL does not work by itself, but it works with this downstream effector called RF6. And there is an effector in between, which is RAL-BP1, that RAL talks to, that allows for its regulation of RF6, suggesting that, these pathways are fairly intricate in how they are controlled. So knowing that this is the case, one of the things that we want to do, which I told you earlier as well, is, is there a way for us to, say, target the RAL-A, RAL-B pathway and regulate membrane trafficking and regulate anchorage independent signaling in cells? Now, during the course of these studies, somewhere in 2014, there was, for the first time, a RAL inhibitor that was identified, which is this compound called BQU57. And interestingly, this inhibitor lacks specificity. Okay, it does not, because RAL-A and RAL-B are very similar, finding something that is going to be selective for one versus the other seems very unlikely, right? And so when the inhibitor was made, it was specific to RAL, but it wasn't able to distinguish between RAL-A and RAL-B. It would inhibit both of them, right? The question then this, gave rise to for us is whether targeting RAL-A but not RAL-B in anchorage independent cancers is realistically possible. Now, why were we thinking about this? We were thinking about this because, so far we had discovered And as far as anchorage dependence is concerned, RAL A seems to be the more prominent player. We also had discovered that GEFs are able to regulate RAL A activation very distinctly as compared to RAL B, not just in normal cells, but in cancer cells as well. So it will be very interesting to see whether targeting only one RAL over the other is as effective or is more effective than targeting both RAL A and RAL B. The problem is, how do you target RAL A without affecting RAL B, right? Now, the interesting way to think about this for us was the fact that in physiology, cells do have a way of doing this. And the way they are able to do this is through this kinase called aurora kinase A. Now, aurora kinase A is interesting because it is able to phosphorylate only serine 194 RAL A. It's not able to phosphorylate RAL B, which means that aurora kinase A could possibly regulate localization or function of RAL A without affecting RAL B in RAS dependent as well as in RAS independent cancers. And the question was, can we prove this? Can we show this regulation is happening, right? Aurora kinases are interesting because, there are three different aurora kinases, A, B, and C. They are particularly relevant in the context of cell cycle regulation, where they play very distinct roles. And aurora kinase A, which is known to phosphorylate RAL A only, is interestingly a very druggable candidate, right? So there is a really nice potent inhibitor of aurora kinase A called MLN8237. Right, and it's very potent. It's able to inhibit aurora kinase A very specifically, but it is very difficult to use because alisertib, which is MLN8237, is poorly soluble in H2O. It doesn't dissolve in water, which affects its potency and specificity for aurora kinase A. This is indeed a recurring problem for many other drugs as well. It's not a aurora kinase inhibitor specific problem. That there are a lot of drugs that are very good at what they are trying to do, but their solubility really compromises, how they can be used, right? This then brought us to a very interesting point in these studies, and ICER has a exchange program with University of Iowa, and Catherine here came to us at that point of time as an undergraduate student from the University of Iowa to do a 3-month project with us. She had a major in chemistry and in biology and was interested in trying to bring together chemistry and biology. And so Catherine came up with this very simple thought or idea that, now that we have a drug molecule that works really well, and we have a, aurora kinase that we really want to target, can we not develop a method of delivering this drug through a nanovesicle or a nanoparticle to allow for it to get into cells and target aurora kinase. Right. So what Catherine started, Ruchira and Sonali picked up, and all of this work was done in this very excellent, collaboration that we've had over the last 10 years with Professor Jayakannan's lab in chemistry in ICER Pune. Catherine was the first person. Catherine and I went and spoke to JK about this. And JK said, you know what, we've been thinking about this problem in the context of other drugs as well. And so, we may have a solution for you where we can make these, drug dextran nanovesicles, in which the water-insoluble drug, like for example camptothecin, that they were interested in, could be trapped in the membrane. And interestingly, he said we could put, a fluorescent dye or something that is soluble inside, the nanovesicle, such that the drug is on the membrane, this dye is on the inside. We can watch where things go inside the cell, and we can also, see what effect does the drug have, right? So we decided to use this method, to make them more soluble, more stable, and more deliverable. These nanovesicles, they manufactured, or they synthesized, and they confirmed the architecture of these nanovesicles. And we in turn took them and added them into cells, right? And when we put them in cells, what we noticed is that there is this very robust inhibition of aurora kinase A activation, without it actually affecting aurora kinase B. Now, the reason it's not affecting aurora kinase B is because that's what the drug is meant to do, provided we give it at concentrations, and get it into cells at concentrations, that keep its specificity. And because we are able to make this nanovesicle and get it into the cell, get the drug into the cell at low concentrations, once you get it in, you have fairly high specificity as far as inhibition is concerned. This, we noticed, reflects in differential uptake, which means in comparison to normal, mouse fibroblasts here or other normal cell types, cancer cell types were able to take the nanovesicle in better as well. This is the same study looking at the uptake of not just rhodamine containing nanovesicle, but now rhodamine containing nanovesicle with the drug as well, right? And so the drug gets in beautifully into cells and is able to work really well there. Now, the question was, once it gets into cells, does it target RAL-A downstream? And interestingly, it does. If you give the aurora kinase inhibitor, and you're able to block aurora kinase A activation, you're able to block RAL-A activation. And it does not do anything to RAL-B activation. And this remarkably also suppresses the anchorage independent growth of these cells. So, and we made a comparison between this nanovesicle containing RAL-A specific drug and the inhibitor that had been made some time back, this BQU47 that targets RAL-A and RAL-B. This inhibition we found was actually better than BQU47, which told us that not targeting A and B together with a drug could actually be beneficial in how these drugs work on cancer cell lines, and how they affect their growth per se. We published this a while back, showing that indeed this is how this pathway works, and that we are able to, suppress anchorage independent growth. What we really wanted to be able to do is to take these nanovesicles, give them in mice with tumors, and see if we are able to suppress the tumors, right? So we said, okay, that's the next thing to do. Can we target tumors in mice with this? It's when we started evaluating that possibility that is when we realized that there is a challenge in being able to do so. And the challenge was that the loading of this drug in these nanovesicles was really low. It was fantastic to be able to get it into cells and do that analysis and show that in principle this can work. But if you have drug loading that is this low, then the challenge is that you may not be able to, give enough of the drug in tumors to be able to suppress tumor growth. So this was a challenge. And this meant we had to kind of go back to the drawing board and see if we can design something that, will allow for this to happen. So at that time, JK and I have had a few students who had worked on these collaborative projects, and Kajal came as a graduate student to the lab, who wanted to work at the interface of chemistry and biology. So JK and I were both joint guides for her during her PhD, and she designed and developed with help from JK's lab these star unimolecular myceles. They call them UMM nanoparticles, designed in a way to allow for the retention of these poorly soluble drugs like MLN 8237. It gets very beautifully trapped in these nanoparticles, and she's able to characterize these. But the really interesting thing that we wanted to ask was, what kind of loading are we able to get? And we were able to achieve significantly higher loading. 4.8%. In this case of MLN is probably as high a loading as has ever been achieved with a nanoparticle for this particular drug, right? So this said, fantastic. We now have a nanoparticle that has the drug, in large amounts as we required. Now let's go back and check if things like cellular uptake are still working. So she made this rhodamine containing UMM nanoparticle, and we checked to see whether its uptake is happening, and its uptake, it gets in very nicely into cells as expected, both in RAS independent and RAS dependent cancers. And now, Kajal was comparing these two to also get a sense of whether behavior is any different. Right. And she then asked whether this treatment is able to affect, aurora kinase A activation and RAL-A activation. And interestingly, what she observed is that the effect of this aurora kinase A inhibition is seen largely in these RAS independent cancers where, RAL-A activation is suppressed very beautifully, but not in the RAS dependent cancers, right? Um, However, when she went ahead and looked at RAL activation, she sees a very distinct effect on both RAS independent and RAS dependent cancers, where RAL A activity is suppressed, but not RAL B. Right. So this told her that, this ability to target rally could indeed be something that these cells, could utilize. She went ahead and looked at their ability to suppress and encourage independent growth, and notice that there is an effect very nicely seen in RAS independent cancers. There is an effect in RAS dependent cancers as well, but it wasn't as prominent as compared to RAS dependent cancers, and this is something that she now is looking at more carefully. But because we had the drug in loaded in these nanoparticles in large enough concentrations, and now we knew that there was an effect, Kajal decided it is still worth going and asking what it could do to tumors, right? So among the things she wanted to do was to be able to ask one, if you put this nanoparticle in mice which have tumors, where does it go? Does it go to the tumors? Does it localize where we are expecting it to localize? What does it do to the effect? Does it affect tumor progression or regression at all? To do this, she made this nanoparticle, but now with an IR dye. And just as we've done with the with the drug, with rhodamine, now she made with an IR dye, and this was largely to be able to see this in mice directly, right? And so when so she does this experiment to try and put these she makes tumors, and then she gives the IR dye, and she's asking where does the IR dye go by itself, and where does the IR dye go, when you compare it to the nanoparticle containing IR dye. And the striking thing for her was that, with time, the IR dye contained in the nanoparticle goes very nicely and stays localized in the tumor as compared to, PBS only control or the free IR dye. And that's seen both in SKVO3 tumors, which is RAS independent, and Miyapaka tumors, which are RAS dependent as well, right? She then pulled out the different organs and looked at the tumors and saw that, most of the accumulation is happening in tumors, and there is a distinct difference in the amount of dye that is present when you give free IR versus IR in the nanoparticle, which suggests that the nanoparticle is being retained in the tumors, that is allowing for the dye to be retained there as well. So we then went on to compare the effect of the drug. And the way this experiment was done is you make the tumors in mice, and then after the tumors have gotten to a certain size, you administer the drug and ask whether the drug is now able to reverse the growth of the tumors. And this is largely, this kind of regression of tumor study is very vital, because it also is something that if you are interested in a drug, you want these to work in these kind of systems. It's unlikely that you're taking a drug, to prevent cancer from happening. Most drugs which are against the cancer are administered, after you have detected the tumor. So to be able to see the tumor and then see if it regresses is what we wanted. We were looking at DMSO only, the nanoparticle only, MLN by itself, and MLN as part of the nanoparticle, to see, how their relative effects compare. And what we saw was really interesting and striking, that one, the nanoparticle was the most effective, containing the drug, was the most effective in reversing, the growth of the tumor. This blue line at the bottom, both in Miyapaka2 and SKBO3 suggests that to be the case. There is also a distinct difference in the effectiveness with which the nanoparticle works as compared to MLN. This free MLN, this is more prominent in the Miyapaka2 cells as compared to SKBO3. And if you go back and remember how they did for anchorage independent growth, SKBO3, the inhibition of anchorage independent growth was distinctly better than Miyapaka. But in mice tumors, the effectiveness against the tumor in mice against Miyapaka tumors, which is pancreatic cancer tumors, are way more effective. This also says that there could be things that are happening in the physiological milieu of how the tumors are formed that contributes to how these drugs are working, and also how the nanoparticle works under these circumstances. We also looked at, in the tumors now, if you take the tumor out and look at aurora kinase or RAL inhibition, what happens, and what we see is that there is, in the presence of the nanoparticle, there is a very strong inhibition of aurora kinase A accompanied by an inhibition of RAL-A, while aurora kinase B is not affected, and that happens in both SKBO3 as well as Miyapaka cells, suggesting that that pathway that we were interested in does seem to be targeted in these cancers to allow for this suppression to take place. So to kind of summarize what I have told you so far, we are now looking at this regulatory pathway that begins from integrins, through RAL, RGL1 goes to regulate RAL-A, and through the RAL BP1 RNO pathway that I did not talk to you about today, regulate R6 activation, which then controls membrane trafficking and anchorage dependent signaling. And RAL-A, interestingly, is regulated by aurora kinase A, both downstream of oncogenic RAS and independent of oncogenic RAS as well. making it a very interesting candidate for targeting with MLN8237 to target RAL activation and encourage independent signaling and growth, right? This now study was published this year in 2025 and is just out a couple of months ago. Um, I also wanted to kind of allude to, the kind of timelines that we are looking at, in doing this kind of work. This work began in 2012, and it's taken us almost 10 years to get to being able to show that this particular pathway and its targeting can be an effective way of suppressing tumors in mice. It's the kind of systematic approach that you really want to take to building a treatment strategy. It's also told us a lot of interesting things about how we want to design these nanovesicles or nanoparticles, how we want the drug to be kept in them and delivered that could improve effectiveness. There are many interesting questions that we are now trying to work on. And one of them is that, now that we know that there is this nano particle that is able to suppress tumor growth, we want to see if we can combine the aurora kinase inhibition with other known conventional strategies of drugs that are, used to target RAS independent or RAS dependent cancers, and ask if, having an aurora inhibitor, does it improve the efficacy with which a doxorubicin or a camptothecin may work? A combination strategy, we think, could still be the best way to take this forward. Eventually, of course, what we are thinking is, we want to be able to take this to, if we are able to get significantly prominent results in drug combinations, to be able to take this to human trials as well. We have another graduate student who's now working on taking this forward, and developing a dual targeting strategy, for aurora kinase and other pathways to test how that could work in these circumstances. What it's also let us do is something that I will go back and show you very quickly, is that it's allowed us to use this as a tool to evaluate, pathways and processes that we have been interested in. And this is actually a very interesting, when I put aurora kinase A here, we are now looking at the, we know that aurora kinase is able to regulate RALA phosphorylation, to regulate early cell spreading. Adhesion is able to regulate aurora kinase A, which is why we are able to target that particular pathway. What we are also looking at is the prospect that aurora kinase A could also regulate RGL1. So along with this pathway, maybe there is a role here too that could drive, this cellular signaling. So we are able to use these nanoparticles not just as interesting drug candidates, but also be able to look at them as tools for evaluating aurora and RAL signaling in cells per se, right? As I said, a lot of this work would not be possible without the funding that we've had from so many different sources, and that's what's allowed us to, use this kind of long-term strategy, to try and build an understanding and a tool, that allows us to now explore how, we can take this forward to a really interesting, drug candidate per se. I'll stop here and maybe take questions, because there is a lot I suspect that I haven't said, which is kind of intentional as well here.
Thank you, Professor Nagraj, for your illuminating talk. First, before we wrap up the whole session, now I would like to request all the participants to post their questions so that the platform really gets its true meaning from his illuminating work. What you have understood and what you really want to know, please ask questions so that it becomes a knowledge base platform.
Yeah, if you have questions, please go ahead. Yeah.
What I could see that his total work has been can be divided into two better parts. One is the pharmacology of the MLN823s, that is its pore solubility and non-specificity, side by side the nanoparticulated drug delivery system for the RAL1 activation. That was really illuminating. So based on these observations, we can welcome questions. So if there is no questions, we can extend our gratitude to Professor
Thank you.
Nagaraj.
Thank you. Thank you.
Yeah, definitely. It was a really illuminating talk, sir, and the way you represented the whole work in a very poised and superfluous manner, it was really wonderful to hear. I
Thank you.
extend my
Thank you.
sincere gratitude to Professor Nagaraj Balasubramaniam for an enlightening presentation. Your work clearly demonstrates how targeting aurora kinase to modulate RAL1 relay activation offers a powerful approach for treating both RAS-dependent and RAS-independent cancers. You refer to overcome MLN8237's poor solubility by developing the NPMLN, nanoparticulate matter, with the drug is truly commendable. Your findings on aurora kinase inhibition and growth suppression in both cell lines are really impressive. Enhanced uptake, tumor targeting, and significant xenograft regression clearly demonstrate an NPMLN strong therapeutic potential. We look forward more commendable jobs and commendable research from your laboratory so that ultimately these big menace in humankind ultimately gets it's we get relief from these peculiar conditions. Thank you once again, sir, for sharing your valuable research and insights with us. Your contribution and your lecture has really enhanced today's academic session.
Thank you. Thank you so much. And if you have questions, please write to me as well. The email ID is adhesionlab@gmail.com. Don't hesitate
Yeah,
to
yeah,
reach
that
out
will be a
to that.
fantastic approach from your side if they can directly have
Yeah,
a
yeah, absolutely.
interaction
Absolutely.
with you over mail,
Yeah,
because in
yeah.
this short period of time
Yeah, I understand. I
some
understand. I
questions
understand.
really come up and some questions remain unanswered.
Sure,
So if it becomes in a truly lucid manner, it becomes easier for both of us.
sure, sure,
So
sure.
can we wrap up with your due permission, sir?
Thank you.
Um,
Yes, absolutely. Yeah.
okay, thanks a lot. I'm wrapping up
Thank
the
you.
whole
Thank you,
session.
everybody. Yeah.
Have a nice day. Today's session is over, and thank you all participants for your active participation. Today, let's call it a day. Thank you all.
Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you.
Okay, from
Thank you,
Professor
sir.
Gulshan Lal. হ্যাঁ, আমি কেমিস্ট্রিতে আছি, চুন